This is the internationally accepted symbol of the male sign. How do male gametes, that is, sperms, start to develop? Have you seen this image before? The answer is yes. When we discussed the oogenesis, and we need help. The help is coming from asking questions so that we can extract information from these images. The starting point is here, and let's see where is this word here. Here we have the embryonic disc. The future human being is going to develop from this disc. There is a cavity above this embryonic disc. This cavity is filled with fluid, and it is called amniotic cavity. Don't worry about the name. You will learn it later on because it's going to be repeated. The cavity under the embryonic disc is called the primary yolk sac, and this is a sac filled with fluid for the sake of nutrition of the embryo, the same as the above cavity. Now let's go and look for point 0 in the development. It is in the wall of the yolk sac, near this yellow marked extension of the yolk sac. Is called the allantois, and these cells are coming from the lining of the yolk sac, and they are the starting cells of developing a human body, and they are called primordial germ cells. When we go to the figure to the right, then we can see that these primordial germ cells developing around the allantois, they are not going to stay there. They are going to travel to the genital ridge. We know what is the genital ridge, and we know its details, and we know the beginning structure of this genital ridge. And these cells, the primordial germ cells, will move towards the genital ridge. Through which route? It is going to take the surface of the nearest structure, which is the last part of the developing large intestine, and they travel through the wall of this part of the intestine, along with the structure attaching it to the abdominal wall, which is called mesentery, and they travel and reach the genital ridges, one on the right and one on the left. In week 6 of intrauterine life, and we are looking at the genital ridge, and we can notice that these red areas are due to proliferation, that is growing into and expanding epithelial cells from the yolk sac. And when these cells grow into the genital ridge, they don't go in as a solid mass of cells, but as irregular cords of cells, and these are called primary sex chords. With the help of questions, what is this organ? This is the testis. This is the fully grown, matured testis. it has different parts, and they are quite different from the parts of the ovary we have seen in the previous session. And we would go into details of the structure of the testis the same way we went through the structures of the ovary. What is this image telling me? It is telling me that the testis in its adult position is down at the bottom of the scrotum. What is the scrotum? Scrotum is a pouch of skin in the perineal area ready to receive the testis, because the testis needs lower temperature than the core temperature of the body. What do I mean by the core temperature? The temperature of inside the body, not that of the skin. And why should it be in a more cooler environment? Because this is what development of the sperms require. It requires lower temperature than that of the core of the body. And the testis at the bottom of the scrotum is connected to the body through a cord called Spermatic Chord. This Spermatic Chord contains arteries, veins, nerves, lymphatics, and the vas deferens, the passage of the sperms from the testis to the inside of the pelvis. This spermatic cord passes through the layers of the anterior abdominal wall, and then it gets into the pelvis. Here is the adult testis, and we want to see what is the green colored structure. The green colored structure is the outer cover of the testis, and it is made of dense connective tissue, white in color, and therefore it is called tunica albuginea. Tunica means a layer and albuginea means white. Next is, from this outer shell or the capsule, septa, the green colored structure, go inside the testis, and they create incomplete lobules, and these are made of fibrous tissue extensions of the capsule or the outer layer of the testis. Now, when these septa go in, they don't go all the way, and they create incomplete lobules. The green colored structures are incomplete lobules created by the growth of fibrous septa. These are lobules, and there are 200 to 300 lobules, and each lobule is filled with 1 up to 4 seminiferous tubules. And these seminiferous tubules, they are long, and they are put in a small space, therefore they become convoluted, they become tortuous. Next, now each seminiferous tubule, which is made of a loop first, and the loop is attached to a duct that is going to carry the sperms to the next stage. These are called straight seminiferous tubules. They are part of the seminiferous tubules, but the last part of the seminiferous tubules. Next, these straight seminiferous tubules collecting sperms from the different lobules. They open into a network of passages in the middle of the testis, and these are called reti testis. Next is we want to take the sperms out of this mesh or network of passages, and this is done by a group of straight ducts, several of them, and they drain the sperms from the reti testis, and these are called efferent ductules. There are 12 to 20 of them. What does efferent mean? It means something going out of. What is the next stage? If we look at this image here, we can see that there is a C-shaped structure on the medial side of, or on the left side of the testis, and the first part of it, which is receiving the efferent ducts, is marked green. This C-shaped structure, which is made of a single tubule, which is 6 meters long and very much convoluted so that it can be packed in a little space. This structure is called Epidermis, and the green part is the head of the Epidermis. It begins at the top end of the testis. Next, we can notice that the middle part of the epidermis is marked green. Therefore, this is the body of the epidermis. And the epidermis ends near the inferior part of the testis, and this part is marked green, and it is the tail of the epidermis. Where is the tail of the epidermis going to lead us? It is going to lead us to a tube or a tubule, single one, which is called the vas deferens or ductus deferens, and it is going to take the sperms away from all these structures, and it's going to join the spermatic cord we have just mentioned. And this ductus deferens is going to the spermatic cord, and the spermatic cord is going to pass through the anterior abdominal wall muscles to get into the pelvis, and the vas deferens is going to have minor additions of a gland called seminal vesicles, and then they end up into ejaculatory ducts, 2 of them, and then they empty into the prostatic urethra. This diagram of the testis, the epidermis, and the different parts of the details of the testis. This figure is left to you to identify these structures, and it is a good exercise to confirm and understand the structure of the testis. What is this diagram showing us? It is showing us the lobules with the loop-shaped seminiferous tubules, and between these 2 lunar shapes there is a single seminiferous tubule, and this is where we are going to look into in details. This is a true microscopic section of a seminiferous tubule. The sections are packed together. Why are they packed together? Because the seminiferous tubule, a single one, is much longer than the space it is occupying. Therefore, it becomes very much packed, and therefore we see these cross sections of seminiferous tubules, one very much compact with the other. Each seminiferous tubule will have a lumen, so that the mature sperms will be traveling through these lumens. And there is a thickness of cells, thickness of the wall. This wall, the thick wall, is made of, from the beginning germ cell, the spermatogonia, to the early formation of spermatids, and this layer is called germinal epithelium. Why is it germinal? Because it is containing cells that are dividing, and we need cells that secrete the hormone testosterone. Now, from where is testosterone secreted? It is secreted from cells between seminiferous tubules, not inside the seminiferous tubules, and these called Leydig cells. Each seminiferous tubule has a wall called myoid cells that will make these walls. This is a closer look, a higher power look at a seminiferous tubule section. And what do we see? We see the myoid cells making the wall of the seminiferous tubules, or to be more precise, the outer shell of the seminiferous tubules, and the thickness of the cells. They are the germinal epithelial cells, and there is a lumen in the center to drain the formed sperms. What is this section? This is a section of the testis, a closer look at the seminiferous tubules, but at the periphery of the testis, because we can see this outer layer, which is called tunica albuginea. Tunica means layer, and albuginea means white. And inside a cross section of a seminiferous tubule, we can see the outer shell, and we can see the germinal epithelium, and we can see a lumen as well. Now we will have a closer look at the germinal layer, the cell that are in this layer, which tell the story of the beginning of the spermatogonia up to the formation of spermatids and sperms. The story starts with the wall of each seminiferous tubule, which contains smooth muscles, fibrous tissue, and blood vessels and other types of cells. This is going to surround the section of the seminiferous tubule. Then at the periphery, if you look down to your left, you will find cells near the periphery. These are small cells with dense nucleus. These cells are spermatogonia, like oogonia, the first stage of development. Then the next stage is this group of cells starting at the bottom. They are larger, and this last cell, you can find that the chromosomes are dispersed because these cells are dividing very rapidly, and they are next to the spermatogonia, and they are the primary spermatocytes. These primary spermatocytes will develop into secondary spermatocytes, and these secondary spermatocytes are 1N cells. They contain half of the number of chromosomes, and they are difficult to see because they divide very quickly. The process of division of cells producing sperms is very fast because the process needs to produce millions of sperms. Therefore, the activity of mitosis and meiosis is very energetic. Then we have a story. This story is that of the spermatogonia, primary and secondary spermatocytes. They need feeding, and this feeding as a source of energy and taking care of is provided by a large cell. This large cell is called Sertoli cell or interstitial cell. This is the nucleus of this cell is faint and large, and therefore it can be recognized very easily. Now look for the circles which would appear now. This is a nucleus of Sertoli cells, and if you look up and to the right, you can see two more nuclei. Then secondary spermatocytes are going to develop into spermatids, the early stage of mature sperms. The nuclei of these cells is near to the lumen. They are small and condensed, having dark color. These are two nuclei of two spermatids. These are another two nuclei of spermatids. This is a much closer look at cells making the germinal epithelium of the testis, of the seminiferous tubules, and we really need questions to find our way through these cells. What are these cells? These cells include the series of spermatogenesis, beginning from the primitive cell, which is the spermatogonia, and ending with the spermatids. Now, here is the wall, the myoid wall. Myoid means muscle, and it is made of smooth muscles and other cells as well. Here is the very beginning cells, the spermatogonia, and they are recognized as such because, first of all, they are near the myoid membrane, the outer shell, and they contain a dense nucleus. So, near the periphery, dense nucleus are indicative of spermatogonia. Here is one, and here is another, and here is a third. These are spermatogonia. You do not find spermatogonia near the lumen. They are always further away from the lumen. Then the spermatogonia are going to develop into the next stage, which is primary spermatocytes. And this is one spermatocyte, and it is a primary because these cells are dividing by mitosis continuously, because we need millions of sperms. We don't need few, we need millions, and to get millions of cells you have to have very fast mitosis. So most of the time they are caught in a stage of mitosis, and they are next to the spermatogonium. Here is another primary spermatocyte, and here is a neighboring one, and these are primary spermatocytes for the following reasons. They are next to the spermatogonia, and they are mostly in a stage of mitosis. The next stage is secondary spermatocytes. These secondary spermatocytes, they are difficult to find, and we are lucky to find one in this section, and because it is going through a very quick division. Therefore, it is difficult to find. And next, these secondary spermatocytes are going to develop into something nearer to final shape of the sperm. It is called spermatids. Why are these cells spermatids? Because they have condensed small nucleus, and they are the nearest to the lumen. Here is another spermatid, and here is a pair of spermatids. Now there's a strange story here, and we look at this nucleus. It does not belong to the process of spermatogenesis. It is pale with a nucleolus. Why this story here is different? Because, first of all, these cells are large. They extend from the outer shell down to the lumen. They are through and through the wall of the germinal epithelium, and they are called the nursing cells. The spermatogenesis cells, they get attached to this cell, and they feed, and they are nursed by these cells. These are called Sertoli cells. Now we need cells to produce testosterone. These cells are outside the seminiferous tubules in between them. And these are large cells, and they contain, of course, granules, and they are called Leydig cells. This is a diagram of the germinal epithelium of the testis, and it looks crowded. Therefore we need questions to find our way into the information of this diagram. First of all, there is a basal lamina, basement membrane. Any epithelial cell sits on a membrane called basement membrane or basal lamina, and you can see this arrow extending from the basal lamina to the lumen, and this large cell is the Sertoli cell or the nursing cell. It is placed through and through the germinal epithelium, and it is a large cell, and its nucleus is large as well. And compare the difference between this blue nucleus and the nuclei around it. You find it that it is the largest, and we have seen that in the cross section of the seminiferous tubules. Then it is the spermatogenesis, and the spermatogonia sitting at the periphery. It develops into the primary spermatocytes, and whenever you see a mitotic figure, then this is a primary spermatocyte. And then secondary spermatocytes are going to be nearer to the lumen, and secondary spermatocytes, although they are difficult to see because they are running a very fast meiosis, and you can see the density of the chromosomes is different from the density of the primary spermatocytes. And then we have the early spermatids, the very early beginning of forming the sperms. And spermatids, they have to change shape, and through a process of changing the appearance and the structure, which is called spermiogenesis. Now, if you come down the slide and the diagram, you can see that there are cells that are called interstitial cells or cells of Leydig. These are the testosterone secreting hormone, and this hormone is picked up by capillaries. What is this section? Now we know this. We have been repeating this several times. It is a cross-section of the seminiferous tubule showing the germinal epithelium. If we look at the lower part, we can see inside this black circle a large nucleus with a small nucleolus. It is nothing like other cells. It is very special, and therefore this is a nucleus of Sertoli cell. This is the nursing cell that will nurse all other spermatogenesis cells. If I ask you what is this section, I'm sure now you can say what it is. And the next is, what is this cell inside this black oval shape? It is nearer to the myoid membrane, the outer shell, and it is having dense nucleus. Look around the other nuclei, you don't see as dense nucleus as this, and therefore this is surely spermatogonia. What is spermatogonia? It is stage number 1 after the primitive germ cells that is going to develop into mature sperms after several stages. After understanding of any subject, things become much easier. You know this section, you know where it is, and you are going to know which cell is this. It is next to the first stage of spermatogenesis, and it is almost all the time in a stage of mitosis. It is running a very fast mitosis, and if you look around, go to your right and you can see a row of this cell. This is the primary spermatocyte. With the help of questions, what is this view? First of all, what can I see? I can see that in the lower part of the slide there is a cross-section of a seminiferous tubule. Then I go to the right, I can see another one, and when I go up near the question mark, then there is a third section of seminiferous tubule, and then I can notice very clearly that there are two cells, large ones, between seminiferous tubules, and these are hormone secreting cells, and they are the Leydig cells or interstitial cells. This is a repeated slide, but this is not for me to comment on, but it is for you to test your knowledge. You see, every now and then you have to test yourself, to test the truth of how much you know. Now, please ask yourself, are you knowledgeable enough to read this slide? Study the nuclei, study their relation to the myoid or the outer shell, study the size of nuclei, and I am sure now you can sail through this slide easily. Now, the story starts as spermatogonia and then goes through primary spermatocytes, secondary spermatocytes, and then spermatid. Spermatid is found near the lumen, and they have the very initial shape, and they need to change to a shape that is capable of, first of all, migration through the male genital system, and then migration through the female genital system. This process of change of shape of spermatid into a sperm is called Spermiogenesis. Now, if you look at figure 1, you will find a round cell with a nucleus, And the nucleus is very important because it holds the genetic material, the 1N group of chromosomes. Now, and in addition to that, it contains some cellular organelles. These cellular organelles are important because we need to produce a structure called acrosome, and it starts, and it is made of enzymes. Enzymes are proteins, so we need to form an initial vesicle that is going to form the acrosome. And if you go to figure 2, you will find that the acrosome is beginning to get larger and collect more proteinous enzymes, but then you will find at the bottom of that cell, you will find something called the centriole. The centriole is a structure, is an anchoring structure, usually seen in stages of mitosis and meiosis, because they form the center, and the chromosomes will be pulled towards that centriole. Here we need a moving structure. That moving structure is a flagellum, and the flagellum is a kind of cellular extension attached to the centriole, and this flagellum is the motor of the sperm that will push it forward in the process of migration. If you look at figure 4 and go to the nucleus and go up, you can see that the acrosome is developing more and more, and the flagellum is getting longer. Now, if you go to figure 5, you will find that the acrosome is about to take the final shape on top of the nucleus, the very front end of the head. And if you go down the nucleus, you see that there is a stack of mitochondria. These are very essential because they are the powerhouse that will move the flagellum. Plus, we now have the essential parts of the sperm, the acrosome, the nucleus, the powerhouse of the mitochondria, and the flagellum. Then there is excess of cytoplasm, and this is pinched away and discarded. Then when you look at figure 6, figure 6 is showing the final shape of the sperm. You can find the upper arrow pointing at acrosome. The lower one is the head and the nucleus. Then the third one is the second piece, which is a stack of mitochondria with little cytoplasm, and then the flagellum. This is a group of changes that transform spermatids into spermatozoa. The other name for spermatozoa is sperm. This is a diagram of parts of the mature typical shape of a sperm. Now, it is not good that I spoon feed you. Now, I have given you so many examples of how to read an image because the image is talking to you. So I will leave you to go through, first of all, the main parts of the sperm, the head, the middle piece, and the tail, and then you can go through the details of these parts. it is part of training to correctly read an image. This is a 3D image of the sperm showing the head at the top of which there is the acrosome, and then there's a little constriction called neck, and then the middle piece, and lastly the long flagellum. What is this image? This image is a diagram, and it is showing that the yellow circle is pointing at the testis. Testis is outside the human body. It's under skin, lying in a pouch of skin which is called the scrotum. For the sake of good environment, a cooler one for sperms to develop. Then this is the primary sex organ of the male. Everything else is no more than passages, and there are passages inside the testis, and the first passage to be outside the testis is the epidermis, which is a C-shaped structure, and we looked into details of this epidermis. Then the epidermis, the tail of which is going to join a single duct which is going via the spermatic cord, which is going to get into the abdomen through the anterior abdominal wall, and this is called ductus deferens, or another name of it is vas deferens. And when this spermatic cord gets into the pelvis, it is now in the intra-abdominal part of the abdominal cavity, and then it turns to become posterior to the urinary bladder, and it continues down. Here it is joined by a duct of a gland called seminal vesicle. There are two seminal vesicles, one on the right and one on the left. These seminal vesicles, they add secretions to the seminal fluid in which the sperms are running. You see, from the start, there is always a medium that the sperms are living in, and they swim in, and they are migrating through the help of the flagellum inside this liquid medium. At this stage of seminal vesicles, it is before ejaculation, and there's a need for some addition to this seminal vesicles. Then it reaches the prostatic gland, and inside the prostatic gland, this duct is going to be called the ejaculatory duct, which opens into the urethra. Here is the point where the male genital system joins the urinary system, and the sperms are carried by the urethra to be ejaculated. This is quite different from the female genital system. The female genital system is not connected to the urinary system in any way. This is the point of prostatic urethra coming out of the